We are live. All right, go ahead, Haley.
All right. Hello, Bio 375. So we are here to talk about our Lesson 9 group problems. So let's go ahead and do number 1. So for number 1, we were asked to draw a picture, basically, of E. coli replicating. So I've kind of made one here on this little flashcard. And because E. coli has a circular DNA, I made the fork right here. And so I don't know how well you can see. So I've labeled everything here. So here is the leading strand, and here's the second leading strand, and here are the Okazaki fragments. And then on the ends here is going to be helicase. And then DNA polymerase is here in green and also right here. And then you're going to see the primase right here and here. And the origin is going to be right between the Okazaki fragments and the leading strand. So it's going to be here on the top and here on the bottom. And then, so DNA polymerase 3 is here right in front of the primer on both sides. And I think that's about it. Oh, the RNA primer right here is this little red line, and it's going to go right before the DNA polymerase.
Sweet. Okay, and then question 2. That was it for question 1, right, Haley?
Yep, we're good.
Okay, so question 2. So this one just is a fill in the blank question, and so they're pretty self-explanatory if you read the notes that we got. So A is DNA primase. This is a protein complex that synthesizes primers in eukaryotes. And then DNA A, this protein promotes the separation of the AT-rich sequence within ORIC. Epsilon, this is a subunit of DNA polymerase 3, contains proofreading activity. DNA polymerase 3, this protein synthesizes a leading strand in eukaryotes. DNA primase, this protein displaces DNA polymerase 3 from the chromosomes to terminate replication. And then F is DNA polymerase 1. G is topoisomerase type 2. H is ORS. I is SSBPs. J is DNA ligase. And yeah, so those are just matching. And then we'll go back to you, Haley.
Okay, so for number 3, it was kind of easy because most of these were actually in the problems for the week, the individual problems. So 3A, 5 enzymes that consume ATP are going to be DNA A, DNA helicase, DNA gyrase, DNA primase, and then DNA. And then for B, 5 enzymes that consume DATP are going to be DNA polymerase 3, holoenzyme, and then DNA polymerase 1, polymerase epsilon, and delta. So for C, 5 enzymes that have 3' to 5' exonuclease activity is going to be DNA polymerase 3 and DNA polymerase 1. And then we're going to have polymerase delta and polymerase epsilon again. All right, so for D, we're looking for one enzyme involved in replication that uses DNA to synthesize RNA, and that's going to be RNA polymerase. And so when we're looking for E, an enzyme that uses RNA to synthesize DNA is going to be reverse transcriptase. So it's just kind of flipped around.
Perfect.
All right, on to number 4.
Okay, number 4. Let me switch over here. Okay. So number 4 is we're supposing we can synthesize the DNA in the lab. And then to perform these 4 DNA synthesis reactions, part A through D, we need to determine the sequence of DNA strands synthesized if we use the nucleotides that they give us. So we're given this single strand DNA molecule. And so for A, we suppose that the nucleotides in this reaction include D-ATP, D-CTP, D-GTP, and D-TTP. So those are like all the normal nucleotides that we need. And so the other strand of DNA would just be the strand that matches up with 5 to 3. So it'd be TA, TCG, TCC, T, GG, TA, G, so on and so forth, all the way to the end, just matching up with the other single strand DNA molecule. And then for B, the nucleotides in this reaction include DATP, DCMP, DGTP, and DTTP. And so since we're missing DCTP, it's just DCMP. The nucleotide will stop at the first C that's in the sequence. And so since this sequence is TATC, GTCC, it'll stop at that first C, so it'll just be TAT. And then the nucleotides in this reaction include DATP, DCTP, DGTP, and UTP for C. So since it contains UTP, it will be, instead of the thymine, it will be uracil from the strand that we'd identified in A. And so yeah, it'd be UA, U, CG, U, CC, so on and so forth. And then D, the nucleotides in this reaction include DATP, DCTP, and then just GTP and DTTP. So since there's no DGTP, it's just GTP, it'll stop at the first G in the sequence, sorry. And that will leave us with TATC. And yeah, that's it for question 4.
All right, so on to number 5. So it's just listing the above proteins. So for A, which proteins are found in prokaryotes? And we said DNA polymerase epsilon, DNA polymerase 1, DNA gyrase, DNA primase, and DNA polymerase 3. So for B, which is found in the mouse, so from the mouse, we can assume that mice are eukaryotes. So it's asking which are found in eukaryotes. So it's going to be DNA polymerase epsilon, DNA polymerase 1, DNA gyrase, DNA primase, and then DNA polymerase 3, and as well as DNA A. So for C, which of the proteins is involved in chromosome compaction, we just found telomerase. And for D, which is involved in this initiation stage of DNA replication, that's going to be MCM helicase, because that's the one that has to unwind. So for E, it's kind of a trick question. It asks which is involved in the termination stage of DNA replication, but there aren't any proteins known to be involved in the termination stage. So that's about it.
Cool. All right. Well, that's what we got for you, class. Hope you enjoyed it. Goodbye.